Hello, I'm Martin Blaser. I'm the director of the Center for Advanced Biotechnology in Medicine, known as CABM. Today, I have the pleasure of speaking with CABM resident faculty member, Dr. Céline Gélinas. Céline is the Senior Associate Dean for Research, and she is professor and chair of the Department of Biochemistry and Molecular Biology at Robert Wood Johnson School of Medicine. So, Céline, it's nice to see you.
It's great to see you.
And let's start. Maybe I'd like to ask you, can you tell me a little about your education and your early career before you came to CABM?
Sure. I'm French Canadian, as you might tell from my accent. So I grew up in Quebec, the province of Quebec. I went to public school until about 12, and then I went to boarding school until college. Um, I went to college at the University de Sherbrooke. It's a French-speaking university. I did not speak English at the time, and I was a chemistry major. And I was introduced to research in the summer, first in the biochemistry department and then in microbiology department in subsequent summers. And I really got the bug for research. I really loved it. And so I then decided to do my graduate degree at the medical school at University of Sherbrooke in their microbiology program. And that is where I became introduced to virology and cancer research, working on DNA tumor viruses. And then from there, I came to the US and to do a postdoctoral training at the McArdle Laboratory for Cancer Research at the University of Wisconsin in Madison. Specifically, I came there specifically to work with Nobel laureate Howard Temin, who discovered reverse transcriptase. And this was really an incredible opportunity for me. So in his lab, I worked on retroviruses, studying packaging and generating generation of retroviral vectors. And given my cancer interest, I also studied at the time what was at the time a really intriguing oncogene that led to incredibly rapid fatal lymphoid tumors in animals within days. This gene is called REL, and I was first to report that it could modulate transcription, suggesting that perhaps this might contribute to its ability to cause cancer. And fast forward a few years later, David Baltimore, who had obtained the Nobel Prize with Howard Temin and Renato Dolbecco, Baltimore discovered and cloned a novel transcription factor in B cells that he called NF-kappa B, which turned out, we now know, is actually a family of transcription factor and a very important one. And it turns out that the REL oncogene that we were studying in Howard Temin's lab not only is a member of this family, but actually was the first identified member of this really important family of transcription factors. So this not only supported my own finding on that REL was a transcription factor, but it's also actually the second time that the research of Howard Temin and David Baltimore crossed paths. So eventually, of course, there's been an entire field of NF-kappa B research that has emerged. And so now we now know that it clearly plays really important fundamental roles in immune response, infection, inflammation, stress responses, and is implicated in many, many disease conditions, including cancer.
Sure. So, what brought you to CABM?
Well, what brought me to CABM is I basically was looking throughout the US and throughout Canada, and frankly, CABM was the place that really had, to me, the best scientific environment. The environment at CABM and at Rutgers provided really incredible opportunities to grow. And obviously, CABM was developing as a very collaborative and supportive environment. And the director at the time, Aaron Shakin, also really played an important role in influencing my decision.
Good. So, what are you working on now?
So, well, our long-standing interest is in understanding molecular mechanisms involving cancer. So we've studied whether it's development, progression, or chemoresistance. And so more recently, we've also looked into mechanisms linked to viral persistence. So our approach has been to be focusing on key signaling pathways, and a lot of our work has focused on the NF-kappa B signaling pathway. and then zeroing in on key players in the pathways and studying them as potential therapeutic targets, and then identifying ways to target them. Um,
Great. Anything you're working on right now that's very exciting to you?
so our most recent work actually has been collaborative and stems from our involvement, from our work on NF-kappa B, which, as I mentioned, is key in infection, and also from our work on protein regulation by the ubiquitin proteasome system. And so this work has been looking at signaling pathways and strategies to overcome HIV latency. And so obviously there's been really tremendous progress in controlling HIV using antiretroviral drug therapy, but persistence of the virus in a latent state is still a barrier to eradicating infection. And so using a screen, we've been able to implicate a role for the ubiquitin proteasome system in maintaining latency and suggesting that we might be able to exploit the specificity of the proteasome system to identify perhaps new classes of latency reversing agents. And so this work, as we've been focusing on identifying this specific proteasome pathway, as well as specific particular E3 ligase required for latency, and identifying candidate inhibitors for this pathway. And so we're currently looking into mechanism of action of interesting candidate.
Yeah, great, great. So how do you relate to students and trainees in your research?
Oh, well, I've always had an open door policy, obviously, and it's supplemented with individual meetings and weekly lab meetings and journal clubs. And over time, I've learned that you learn as much from your trainees as they learn from you.
More.
And I've also teach in the graduate school and as well as in the medical students. And I direct a summer research program and a distinction in research program for medical students. So I have a lot of opportunities to
Yeah.
interact with trainees in the
Well,
lab and in the
well,
classroom.
well, you wear a lot of hats because you're also the chair of the Department of Biochemistry. And
Mhm.
how's that been? Are you the first woman to be the chair of that department?
Uh, yes, come to think of it, yes. I never thought of it. I find this very rewarding, both in my role as chair and as research dean, to have the ability to really help facilitate the success of our investigators and my colleagues.
Yeah,
And I also direct the regulatory core for the New Jersey Alliance for Clinical and Translational Science, or NJATS, which is a consortium that's dedicated to the development of new approaches to health problems in New Jersey and translating into therapies and treatments. So it's really an exciting time right now for Rutgers, the medical school, and CABM, I think, with so much growth and opportunities for research across the board.
great. I just want to ask you one more question, and that is, imagine it was the end of your career, and
Mm-hmm.
maybe it's a little morbid, but you had to write your scientific obituary. And so what would you put in your obituary? What are you most pleased or proud of?
Well, I think so. Research certainly has been really fascinating, and it's been a really an exciting journey to conduct research to help elucidate really important mechanisms in cancer and disease. Um, but honestly, I think perhaps what I find the most rewarding has been the opportunity to be able to train and mentor a really wonderful group of students, postdoc, research associates that have gone on to really successful careers. And I'm really proud of their accomplishments and that I was able to guide them on their career paths and foster their growth and success.
Great, great. Thanks very much,
Thanks
Celine.
so
And
much.